We are now going to look at the path of light through the eye and the organization of the retina. On the very least, I'm going to ask you to explain it. In paper tests, I like to ask you to draw it. And we're going to relate this diagram of the retina to histology in lab. We talked about the eye structures in the last section. We talked about the sclera being the tough outer part of the eye here. And we talked about the retina being where all the receptors are as the inner layer, inside there. And we talked about how the cornea is this clear, transparent part on the anterior sixth or eighth of the eye. We talked about how you have an iris here that controls the amount of light through the eye, and it has the lens behind it, right there, lens, that focuses the light on the back of the eye. It is going to first go through the cornea. So here's our light. It's going through the cornea, and that actually refracts it a little bit. So it changes the angle just slightly. And the iris controls how much light is going through. If it is bright light or a near object, the iris is going to be constricted. If it's a far object or dim light, or you're upset, or if you're attracted to something, it's going to be dilated and wide. So the light goes through, gets bent again or refracted by the lens, and it ends up hitting the back of the eye. And notice it got flipped upside down. So here, it got flipped upside down. Now, this person's looking at a 3-leaf clover, I think. So it's going to actually have some tension in these muscles right here. Ciliary bodies or ciliary muscles. And they're going to create the tension so you could focus on a near object. If you're looking at something far away, like this letter J, what's going to happen is those muscles are going to relax a little bit. And again, it will go through, it'll cross, and it will turn upside down on the back. Oops, I completely missed that. Of the eye, so it'll be an upside down and backwards J. There are two types of photoreceptors, and they both face the back of the eye. There are these cone-shaped ones that are called cones, and there are rod-shaped ones that are called rods. Now the rods are towards the outer edges of the retina. So if this is the eye, cornea there, we have the lens, the retina would be back here. The rods are going to be here. It's easy to draw rods, and here. So they're towards the periphery. In the center area, we're going to learn is only cones. Now these rods, they have these photoactive pigments that are going to actually deactivate the rod when it's struck. Now the rods are attached to these bipolar cells. Remember, bipolar neurons are rare. So it's attached to these bipolar cells. And those, in turn, are attached to a ganglion cell. They're called ganglion cells because they were cell bodies they thought were outside the central nervous system. Spoiler, they're in the central nervous system. So, basically, light hits the rod, signal goes to the bipolar cell, signal goes to the ganglion cell. But look! 3 rods, 1, 2, 3, are converging on one bipolar cell. Several of these bipolar cells are actually going to converge on one ganglion cell. So there is convergence, meaning you actually don't know what your rods are showing you. Your rods are making up what they think they see. And there's some very cool optical illusions based on that. Cones let you see color. There are at least 3 types of cones. There's been argument that there's a 4th type that some people have. Rods actually let you see a purple tone of light, so I'm not completely sure about that. But there are red cones, there are green cones, and there are blue cones. Notice, no yellow, no orange. What happens when you see colors in between is the green and the blue rods are both excited, one's excited a little bit more, and you see that as a purple. Or yellow and orange, the green and the red are both excited, one is excited a little bit more. So cones let you see different color, and they're sensitive to different wavelengths. In the fova centralis right here, there are only cones. So light is going directly in and hitting the fova. So when you're looking directly at something, you should see super clear vision because you only have cones in the center, whereas the rods are out here in the periphery. Rods are on the periphery, and they're converging on bipolar cells. So here's 3 rods converging on one bipolar cell, and let's just draw another bipolar cell for the sake of argument. That's going to converge on the ganglia cell. They're on the periphery. They're sort of making up that, oh, there's something there, like a black and white crown Victoria. They're just motion detectors. They're letting you know there's something on the periphery. And that's actually why your dog and your cat will go crazy for a laser pointer. They have mostly rods and cones for night vision. So they see the laser pointer, they think it's movement, and they pounce. Now, we're visual creatures, and we have a lot of cones right here in the center, in the fova centralis, and cones don't converge. So each cone is attached to one bipolar cell. Each bipolar cell is attached to one ganglia cell. So you get a lot of detail looking straight. Let's say a histology slide of the retina later on today, maybe. Now they need bright light to function. And yes, I spelled light wrong there. So they need bright light to function. And that's why you don't have very clear vision when it's dim. Now the other fun fact about rods is in dim light, they take about 20 minutes to actually really get going. So if you ever want to go stargazing, it needs to be completely dark outside, and you actually need to wait a little bit before you see everything in the night sky. I highly recommend going to Greece for this, by the way. So, A is rods, B is cones. Which one's for day vision? Which one works in bright light? Correct, it is B, cones. Found mostly on the outside of the retina, the periphery. Correct, A, rods. Which one is found in the fova centralis? Correct, it is B. Night vision. A. At least 3 different types. Correct, cones. It is blue, green, and yellow.